Junyue Cao. Junyue Cao is an assistant professor and lab head of single-cell genomics and population dynamics at Rockefeller University. His work focuses on developing novel high-throughput single-cell genomic profiling techniques by combinatorial indexing. These techniques enable profiling millions of single-cell transcriptomes from entire organisms in species including worm, mouse, and human. Okay.
Okay, thank you for the introduction. Okay, so let me check if I can share the screen. Okay, great. So thank you for the invitation. I'm Jun. I'm a new assistant professor and the lab head of the Rockefeller University. So today I want to share with you our previous and current research about global organism-scale views of cell state heterogeneity and dynamics by sci-RNA-seq. Okay, so the question we are interested is to understand if it's a life generation process. So as we know that we each are generated from a tiny single cell, and this tiny single cell continue to divide and differentiate into many different cell types. They form different tissues, organs, and finally our body. So because cell is a basic function unit, so we can imagine to understand the life generation process, we need to comprehensively characterize every single cell state and also how the dynamic change during the life generation process. So the first question is then how to characterize the different cell state. So traditionally, we use some cell marker-based approaches to separate cells into different cell states. But you can see here, the cell marker-based approaches can be highly biased based on which marker we use. So more recently, people use single-cell genomics or most by single-cell RNA sequencing to characterize the different cell types based on hundreds to thousands of gene expression. So in this way, we can separate different cell types with less bias. But there's a key limitation for single-cell genomics. So for example, here, if you just check the cell number in the neuron system, here we have hundreds to thousands to even millions to even billion cells in the real biological system. But the throughput of current single-cell genomics are mostly limited to like several thousand cells in a single experiment. So, and this is make it very challenging, for example, to identify some rare cell types or to compare the cell state changes across many different samples. So our first goal is to develop a high throughput single cell genomic approach to get over this limitation. So the key here is a strategy called combinatorial indexing. So combinatorial indexing is a strategy that enables single cell detection without physically isolating a single cell. So here is how it works. For example, here we start with a group of cells, and we want to detect the single cell molecular information. So for the first step, we split the cells into a multiple well plate. And for each well, actually, we have tens to thousands cells. And then we can barcode the molecules in each cell with a well-specific barcode. Right, you can see here, for example, the cells in this well, they will receive the same barcode. And then we will pull all the cells. And then we repeat this whole process. For example, you can imagine after several rounds of random distribution, indexing, and pooling process, the molecules in each cell will be barcoded by a specific array of barcode. So the key here is that because here we do the barcoding inside the cell without disrupting the cell, which means that the molecules from the same cell, they will receive the same barcode array. So then we can directly disrupt all the cells, extract the molecules for sequencing, and based on this barcode array, we can computationally group molecules from the same cell without isolating a single cell. So as you can see here, one key feature of this strategy is that the detection capacity or the number of barcodes we can barcode the cells exponentially increase with more round indexing. For example, with 2-round indexing, we can already barcode like 40,000 cells in a single experiment. And with just another round indexing, we can increase the throughput to another like over 100-fold to million cells in a single experiment. So based on this strategy, here we developed sci-RNA-seq or single cell combinatorial indexing RNA-seq. So the strategy is very straightforward. Here we have a group of fixed cells or nuclei, and then we can split the cells into a multiple well plate for the first round indexing by indexed reverse transcription with barcoded oligo-DT primers. And then we can pull all the cells, redistribute the cells to a second plate for the second round indexing by PCR. And then we can pull all the cells for sequencing based on this just two barcoding combination. We can identify single cell gene expression. So to test if this strategy is working, here we first test it to generate a cell atlas of entire organism like the worm. So here we use the worm as a model system because we already know the major cell types from the system so that we can use this as a control. To see that, if we can use single cell RNA-seq alone to characterize all the cell types from the entire organism. So, and you can see here from the data, we profiled around 50,000 cells. So each worm has around 1,000 cells. So this is around 50x cellular coverage from the entire organism. And from here, by cluster analysis, we can readily identify all major cell types from the worm, from the body of muscle to germinalized cells to intestines and others. And we can also further characterize the different cell type and the cell type specific gene features. And from these gene features, we can further purify each cell type for further study. So in summary here, I show you the strategy for combinatorial indexing and how we can use this to characterize the cell type heterogeneity across the entire organism. So it turns out that this technique, the sci-RNA-seq, is not just a single cell RNA-seq technique. It can be used as a platform so that we can like co-barcode or co-profile other molecular information in the same assay. For example, here we develop another technique called sci-CAR so that we can barcode the gene expression and the chromatin accessibility from the same cell and to study their covariance across tens of thousands of cells. So if you are interested, please check this manuscript. So in summary here, we can use the combinatorial indexing strategy to barcode single cell gene expression or other molecular layers so that we can use this to characterize cell state heterogeneity. So the question is that for these different cell types, how they are generated in development, or how can we characterize the cell population dynamics to understand how these different cell states actually are differentiated in early development. So to answer this question, as you can imagine, we not only need to characterize the cell state from a single developmental stage, like here, a single mouse embryo. Actually, we need to use this to characterize the cell population dynamics across different developmental stages from the early to late developmental stage. But here, the limitation is still the throughput. For example, here, if we check the cell number in mouse embryos, here we have hundreds to thousands cells to even millions of cells in each individual. And this is much higher than the original version of sci-RNA-seq and also other commercial techniques. So to further get over this limitation, here we further improve the sci-RNA-seq strategy to add another round indexing by indexed hairpin ligation. So as I mentioned earlier, with 3-round indexing, we can detect millions of cells in a single experiment. So to test this, here we apply sci-RNA-seq 3 to profile 61 mouse embryos across different developmental stages from 9.5 day to 11.5 day to 13.5 day that span the main organogenesis stages. And from a single experiment, here we can profile over 2 million cells across 61 different individual samples. So what can we learn from this data? So as you can imagine, like other single cell genomic study, by cluster analysis, we can readily identify and annotate the main cell types from the connective tissue progenitors, lens, and neutrophils, like the rare cell types. But because here we have a much larger data set, we have tens of thousands, even hundreds of thousands cells in each cluster, so then we can further zoom in to each cluster. For example, here, if we zoom into the epithelial cells, took these cells for subcluster analysis, and now we can identify more subclusters, and based on the gene markers for each subcluster, we can map these different subclusters to different epithelial cells from, for example, from the epidermis to the like the lung epithelial cells to the epithelial cells in the GI tract and others. So in this way, we can characterize the epithelial cells across different organs in the same study. So this is just one simple example. Here we try the similar strategy to every main cluster we identified here. So here we can identify over 500 subclusters that represent different cell state in embryonic development. So after characterizing this different cell state, the next question is that how this different cell state are generated in development. So to answer this question, here we use another algorithm called UMAP that's also incorporated into the Monaco package. So which can group the cells from related lineages into a single trajectory. For example, this is the cells in the T space colored by the main cell type, as I showed you earlier. And now we can see the cell transition from the T space to the UMAP space. For example, here we can see the related cell types tends to group a single trajectory. For example, here are all the blood cell types. And this group of cells, like this long trajectory, represents the cells from the neural tube and also the neurons that form the central nervous system. And this is the mesenchymal cells that includes the stroma cells, myocytes, chondrocytes, and others. So after this transformation, we can see these 38 main cell types are grouped into 10 major trajectories, from mesenchymal trajectory to epithelial trajectory to different neural crest trajectory to endothelial trajectory. And if we zoom into some specific trajectory, for example, here if we zoom into the neural tube trajectory, and now we can see a smooth cell state transition path from radioglia to developing neurons. And if we rotate the trajectory, and now we can see another path from radioglia to oligodendrocyte. So because each trajectory here actually still have hundreds to thousand cells, so then we can further zoom into specific trajectory to study detailed cell state transition links. For example, here, if we zoom into the epithelial cell trajectory, which is a small cluster here, and we do the thermal analysis, and now we can see the epithelial cells actually group into more complex 3D structures. So here we color the epithelial cells by the main cell types that we annotated before. And also we can compare this result with the same plot by coloring the cells with the different developmental stages. And combining both information, we can readily annotate the epithelial trajectories in different organs. For example, this is a mid-gut hindgut trajectory. And here we can also identify the foregut trajectory, which already separate into two directions, one to the stomach and one to the lung. And we can also identify the epithelial trajectories and form regulates the limb development, for example, the epidermal rich cell trajectory, the renal epithelial trajectory, and others. So in summary here, we can use this to study the cell state differentiation paths during development, like for the epithelial cells. And this is just one example. Actually here, we tried a similar strategy to all the other 9 major trajectories. And in total, we can identify 56 cell state transition trajectories that form all major systems or organs in the mice. So because here, for each cell, we know the developmental stage information for each cell. So from here, we can also use this to infer the cell state transition directions. And we can also use this to characterize the gene dynamics and also key transcription factors that regulate the differentiation for each trajectory and form different organs from at the scale of the entire organism. So in summary here, by development of high throughput single cell genomics, we can use this to characterize the cell lineages that form major organs from entire mammalian embryo. And the next question is that after this embryo actually are generated, how the cell population dynamic change in later developmental stages, for example, in the fetal state. So to answer this question, here we further improve this strategy so that we can use this to profile the cell population, not just in embryo, but also across different human fetal organs. So here we apply this to over 100 human fetal tissue samples from 15 major organs. And in this project, we sequenced around 5 million cells in total. So because for each cell, we know its spatial information, so we can further split the cells into different organs and use a similar trajectory analysis to identify the main cell trajectories that form different human fetal organs. For example, here we can identify the cell trajectories that form the adrenal, the brain, the eye, and also other organs. So we can also use this to characterize some cell state actually that not characterized in previous study, and also validate this by immunostaining data. So what can we learn like from this data set? So because like in this study here, we characterize the cell population across many different tissues so that we can systematically compare the main cell types. For example, here we can readily characterize the function-related surface proteins, secreted proteins that specifically enrich in each cell type. And we can also characterize thousands to non-coding RNAs or key transcription factors that are significantly expressed in each cell type. If we compare this data set with other database, we can also identify like thousands of pathways, phenotype, or disease that significantly linked with each cell type. So here, just want to give you one example. For example, here, if you check the pancreas data, we can identify a cluster of isolate endocrine cells. Actually, if we check this cell cluster, we can see it's already separated into different clusters to different like alpha cells, beta cells, and other endocrine cell type. Like here, if you check the top like enriched transcription factor for this cell type, we can see actually like the top enriched transcription factors have like all been reported to be involved in the differentiation of this cell type. So here, if you check the secreted protein, we can see they are highly related with the cell type specific function. So similarly here, we can also check like identify the cell type specific non-coding RNA related biological pathways. It turned out that a lot of them had been reported, but we also identify a lot of novel links. Okay, so because from this data set, actually, I think a key feature of this is that we process or profile cells across multiple organs using the same technique, the same platform in the same experiment, actually, so that we can group the cells from different organs and easily integrate them all together. For example, if we took out all the cells across different organs for cluster analysis, and now we can see the cells are grouped into different clusters. And here, if we color the cells by the different organ type, we can see a lot of clusters actually include cells from multiple organs. And it turns out that this makes sense. For example, if we color the cells by the main cell types, not by the organ type, for example, here, if we color this group of cells by the main cell type, we can see the different blood cell types actually group into individual clusters, even though they are from different organs. So because here we like have hundreds to thousands blood cells, so then we can further zoom into this group of cells. For example, here, if we just took the blood cells and do the similar trajectory analysis, and now we can easily identify the blood lineage from HSPC to different blood cell types. So, right. So here, if we color the cells not by the blood cell type, but by the different organs, actually, we can see the different blood cell states actually are enriched in different organs. So here, I just want to give you one simple example. For example, here, if we just zoom in to the erythroblast, like this cell type, if we take out this group of cells, and by cluster analysis, we can segment this erythroblast to different developmental stages, from the early progenitors to terminally differentiated cells. And if we just took the one substate, the early progenitors for the erythroblast, and check its distribution across different tissues. For example, in this plot, each dot here represents the cell proportion from an individual tissue. So for example, here we can see the early progenitors of erythroblasts are enriched in the liver and spleen as expected. But here we can also see it's enriched in the adrenal compared with all the other organs. So, and here we further validate this result by immunostaining. Here we do identify clusters of erythroblasts in the fetal adrenal that potentially serve on the minor side of erythropoiesis. And this is further validated in mouse tissues. For example, here, if we took out the mouse adrenal, and if we compare that to the like from the mouse kidney, and here we do see a significant fraction of erythroblasts in the adrenal, and the proportion actually is comparable with the adult marrow. Okay, so this is just one simple example to show you that by high throughput single cell genomics, we can profile, like by profiling a lot of cells across different organs, we can characterize the cell state differences, like for the same cell type across different organs, and we can further zoom into specific cell type or even subtypes to study their gene expression differences or their distribution across different organs. So in summary here, I show you by this technique, we can profile 56 embryonic cell trajectories that form different embryonic organs in mouse embryos. And by applying this to human fetal tissues, here we also characterize 77 fetal cell trajectories that form different human fetal organs. And you can imagine if we can combine these two data sets together, potentially we can construct a cell state transition trajectory from the very early embryonic stage to the late fetal stage. So for this aim, here I use a published cell integration assay to integrate these two data sets together. So here I color the mouse cells by the different developmental stages and human cells by the fetal stage. So integrating both data set, here we can identify already identified some cell smooth transition trajectory from the early embryonic cells to the late fetal stage. So if we color the mouse cells, not by the developmental stage, but by the main cell trajectories that we annotated before. So this also one of the results that human and mouse cells from the same trajectory tend to cluster together. For example, the human and mouse epithelial cells are grouped together here, and the human and mouse mesenchymal cells are grouped here. So if we zoom in to specific trajectory, because here we have if we combine these two data sets, there's over 6 million cells. So that if we zoom into the blood cells, here we have hundreds of thousand cells. And by SEMA analysis, for example, here, we can readily identify the detailed cell state transition trajectory, for example, from the early white blood cell progenitors in the mouse. And we can see it transit to different human cell types, to the lymphoid cells, myeloid cells, and others. So, and from here, we can further characterize the conserved gene features and also transcription factors that in human and mouse. For example, here we can identify 81 conserved genes in erythroblasts in both human and mouse, and also identify the TF that's enriched in both human and mouse erythroblasts. And here, if we check the transcription factors, most of them have been reported to be key for regulating the differentiation of specific cell type, but there's also some novel genes that have been that hadn't been well studied before. Okay, so in summary here, I show you a single cell RNA-seq strategy by combinatorial indexing called sci-RNA-seq, and using this technique, we can use this to characterize the cell population dynamics during mammalian organogenesis and also in human fetal development. So integrating both data sets, we can use this to infer the cell state transition trajectory from early developmental stage to the later fetal stage for understanding the life generation process. So with that, I want to thank the great support from my mentor Jay from the University of Washington Genomic Science Department, and also the great support from our collaborators, especially Trapno Lab, Watson Lab, Illumina, the BDRL in Utah, and Fred Hutch. So also this is a funding source and support this research. So actually, I just started my lab in Rockefeller this August. So I also want to thank the support from my lab members. So if you are interested in single cell genomic technique development, feel free to contact with me. Okay, thank you, and happy to take any question.
Thanks, Junyue. Great talk, and thanks for pushing the single cell genomic technology to another level. Here's one question from NUS, I think. He said, thanks for this great technology. We all know that there is no perfect fixation protocol. I wonder how much of RNA is lost during these processes. And do you think the method might be useful for detecting lowly expressed RNA?
Right, that's a good question. So currently we try the different fixation method like messenger fixation, PFA fixation, and also I think it's also compatible with formaldehyde fixation. So generally for messenger fixed cells or PFA fixed cells, if we use two-level indexing strategy, like for example, the sci-RNA-seq with two-level indexing, we can detect a compatible signal compared with the commercial technique. For example, in a recent paper that we published in Nature Biotech, which is another technique called SkyFate, we use PFA fixed whole cells for sci-RNA-seq with two-level indexing. We can detect 30,000 or 40,000 unique transcript per cell. I think this is similar with other technique like the 10x system. Right, but the problem I think for most of sci-RNA-seq experiment, I think the difference here is that we use PFA fixed nuclei instead of cells. So as you can imagine, there's much less transcripts in the nucleus compared with cells. And also, I think another key for single cell combinatorial indexing RNA-seq is that here actually we permeate cells and do the reaction in situ, which means that we need to generate puncture holes on the cell membrane so that the enzyme can enter the cell. So in this case, the sciRNA is actually sensitive to the external environment. For example, if there's a loss of RNA or protease in the environment, this may also enter the cell and contaminate the cell. So in this case, I think this is a key factor that we should consider for designing experiment or use sci-RNA-seq to profile the different tissues. Right. So does this answer your question?
Great. And so yeah, I have a question. So regarding the post-speed protocol, do you think, because cells are very sticky, do you think the downplay rate is going to be higher in this technology? How to solve the problem?
This is a great question. Actually, during the development and optimization of the sci-RNA-seq with 3-level indexing, we optimized the loss of conditions because the key limitation for the sci-RNA-seq is that we cannot use sort to sort single cells. Instead, we need to dilute the cells into multiple wells. So there are two strategies to reduce the cell clumping. One is to use mild suncation to break the cells into single cell suspension. And the second is that we use very stringent filter step to remove the doublets. Right. I think for now, for sci-RNA-seq rate, the doublet rate is around 5 to 10%. And this is not due to barcode deletion, mostly due to actually the cell cell clumps to each other. But I think this doublet rate is comparable with other commercial techniques.
Okay. Oh, there are many questions now. So will you look into the Q&A table and pick questions?
Let me see. Thanks for the, okay. So I think here's one question. When you annotate cells, how often do you find cells that cannot be annotated? Either a mixture of multiple cell types or potential new cell types? I think this is a great question. So I think it also depends on the system we are profiling. For example, when we are profiling the mouse embryonic cells, so because actually there's not published data set that have comprehensively characterize the different cell state in mouse embryonic development. So it turned out that actually it's very challenging to characterize the different cell state because here we can characterize over 500 different cell state. So in that case, we normally annotate the cells based on its lineage. For example, we can see this different cell state, they are different from each other based on the gene analysis, but it's hard to separate them or to annotate them because actually we don't have a previous name for this. So we normally annotate the cells as the different developmental stages for a specific cell type. For example, for some stroma cells, and we can check the proportion of cells from different developmental stage and use this to annotate the cells and progenitor cells, or the intermediate cell state, or the mature cell state. So, but for the human fetal data, I think it's rather straightforward because currently there's other the mouse cell atlas generated, including the GoLab. So it's easy to use this other atlas data as a reference to annotate the different cell state. But still, we can identify some very rare or very small clusters that we don't know actually where they are. So normally we just annotate these cell clusters by the specific gene markers, and we can also use these gene markers to validate these cell types.